distinguished delegates on and off the dais. At the outset, I thank the organizers of VSI for providing me this opportunity to talk to you about the perspectives of sugarcane breeding and value addition and product diversification as it is happening particularly at India and very specifically at Sugarcane Breeding Institute. That is the action points on the issues facing sugarcane agriculture, particularly in India. So, around 120 countries grow sugarcane, and majorly it is cultivated in the countries like Brazil, India, Thailand, China, Pakistan, Mexico, Indonesia, etc. And coming to specifically the problems in India, that is area is around 5 million hectare, and slightly it is increasing now because of the bioethanol and other opportunities. And our problems, grey areas are mainly in the number of ratoon crops, which is very dismally poor, 1 to 2 ratoons. And the area under mechanical harvesting is very, very poor. Though, of course, with the availability, with the non-availability of labor, factories are moving towards mechanization, particularly very fast in the southern India. So some of the achievements, like about the varietal role, as just now you heard, one variety can change the entire scenario of sugarcane agriculture in the country. And the exports also started picking up from 2016, 2017, as the variety Co238 started spreading. And last year it was very high, around 112 lakh tons, so sugar was the highest agricultural commodity to be exported from India compared to other like milk, meat, or fish, or even rice. Sugar was the highest, led in the commodity under export. And of course about the bioethanol policy, which you heard from the main person itself, the horse's mouth itself, from our honorable minister. And there are so many products, like from food to feed to fertilizer to furniture to farming, that is even biofarming and producing value-added products. And even the edible and inflorescence of Chacra medule is now is a preferred vegetable in many of the islands, especially in the New Guinea and New Caledonian Islands. Now coming to the research priorities, now has the sustainable sugarcane production, as almost all the speakers spoke, and especially for India, because 238 made a revolution, and to sustain that sweet revolution is a very big challenge, and reducing the cost of cultivation, and make sugarcane agriculture more climate resilient and resource efficient, and product diversification. I will be speaking mainly on the breeding challenges, or the challenges how we can overcome through breeding. So first is breeding strategies to improve varieties for sugar, ethanol, dual purpose cane and size specific varieties, so that the productivity can be improved. Then fast tracking and improving precision in breeding. Then good quality breeder seed, as everybody knows the importance of breeder seed, and the varieties for a targeted yield, that is a variety alone cannot do, a combination of agronomic practices which can make that possible for a sustained or an improved yield. And for that it's our industry or in the institutes cannot do it. It is a collaborative approach between the industry and the research institutes. Now, there is a very unique system in India. Of course, all the research institutes have research priorities, but in India, the research, that is breeding, is likely centralized, because there is a facility at Coimbatore, and where all the parental clones are planted, and it's a national facility where 24 stations across the length and breadth of the country participate in breeding program. It's a very unique thing for specific to India. Now coming to the breeding strategies, that breeding superior varieties for different maturity groups as well as location specific varieties. Then enhancement of sugarcane germplasm, that is mainly to address climate resilience and resource use efficiency, and genomic and molecular approaches mainly to improve precision and improve selection, and the gene discovery, that is add a new trait, correct a deficiency in an otherwise promising variety. Then cropping system centered around appropriate varieties. Then enhancing the physiological efficiency as told by Dr. Prakash. Then host resistance, of course, is a dynamic thing because varieties break down to diseases and host resistance need to be improved. and economic and analysis to know both to address the gene-to interaction as well as the impact of varieties, how they are impacting the society, and of course the transfer of technology to the grassroots level. So there is no, I am skipping these slides because you know the importance of code 238. What made us here today probably because of the success of 238 and the higher income the factories and farmers are getting today. And there is no doubt that the person was awarded, that the breeder was, the scientist was conferred with the highest civilian award in the country. And there is another tropical variety, of course, that is 86032, the main variety in Maharashtra. We can see here, you are telling about 80 tons per hectare, 100 tons per hectare, 120 tons per hectare, 324 tons per hectare. Everything has been demonstrated not by one farmer, by thousands of farmers in the Sangli region in Maharashtra, where 325 tons per hectare is a very common thing, and even up to 167 tons per acre per, that is 18 months have been realized. That was the highest yield realized, that is coming around 425 tons. And this has already happened not with one farmer, with many farmers in the state of Maharashtra. So that is the advantage of variety when it is coupled with suitable agronomic practices. Of course, everything should start with an improved high quality seed. Micropropagation with virus indexing will help you to get a clean seed, clean micropropagated seed, and using that in a breeder seed program. You can see in the background, this is a 7-month-old crop from tissue culture plants. So this not only assures a production of high quality breeder seed, but you can see how the seed farmer gets the yield. At 7 months, he is able to get 110 tons per hectare. So his profit, you can imagine the additional earning by these seed farmers. So not only the seed farmers are providing good quality seed, but his own income is also improving this way. So now next major thing is reducing the cost of cultivation. Among all the countries, the cost of cultivation is highest in India. So main strategies are to improve the multi-ratooning potential and also to develop short duration varieties mainly to realize 3 crops for 2 years or for a country sustaining the sugar recovery. And hybrid seed production through true seed. I will be explaining in 1 or 2 slides that. Then mechanization, complete mechanization from seed bed to harvest. And of course, the doubling of farmers' income through integration of technologies. And here the short duration varieties are very important. We have already released one short duration variety, which is as well climate resilient or waterlogging resistant, and which has a very high sucrose content. Of course, it has been released for the peninsular zone, and its suitability to other areas need to be worked out before the variety is spreading to the farmers. Because we get the information of this varietal spread, but it can lead to other problems, so the variety has to be tested in research stations. So now true seed, that is, that's a dream project of our former director, Dr. Bakshi Ram, and that is reducing the seed weight from truck to pocket. We need just 32 grams of seed for covering 1 hectare. That is how to make that seed possible. So we have tried several mechanisms, and in this one, the repeated selfing has been found to be very useful. Repeated selfing, identifying the selves, then molecular profiling, and advancing the best selves. And through a marker-resisted selection, you can go for selecting the best inbreds to make hybrids. And these are the hybrids at the 6th generation inbreds. When we developed, we found a population which is near uniform, more than 90% uniform, showing that true seed-based agriculture is possible. Now we have to refine that technique to reach it to the commercial level. We have also demonstrated that at the 7th generation, there is a high level of inbreeding depression. By crossing to 6th generation, hybrid vigor is obtained. In the case of sugarcane, hybrids look like very uniform and look like the normal sugarcane varieties. That shows that true seed in future that again requires more research to take a true seed in a pocket or in a packed cover and then develop seedlings, seedlings and plant in the field. So futuristically, that is another research we are strongly at. And then climate resilient sugarcane develop varieties, and also for resource use efficiency, not only salinity, waterlogging, drought, diseases, pests, of course for India particularly red rot. Then here there is a need for pre-breeding as well as speed breeding, because pre-breeding is a very slow exercise in sugarcane. It takes 30 years to realize a variety. So here it has to be combined with speed breeding, and of course gene identification and marker development. And for that, you know, India has the highest germplasm collection. International collection is coming to 806 international collection. Apart from that, we made several collections through several expeditions, which is even now in vogue, collection of new types and under different categories of species, exotic hybrids, then we have co-varieties, co-allied, interspecific hybrids, then germplasm utilization line, cytoplasmic diverse line. Everything is accounting to around 7,000 lines available at SBI. And here there are very specific and targeted pre-breeding work efforts going on, mainly to improve the species as such, that is intra-specific improvement of species. Then cytoplasmically diverse, because our hybrids are represented by the officinarum as the female. So we have a spontaneum as well as a Ryanthus as the female parent. Then again, to hitherto unutilized typical officinarum to be mobilized in breeding, and race-specific and germplasm enhancement, mainly for specific characters like red rot and drought, and creation of that is magic population encompassing 8 different sources. And we have a very strong cytological analysis by which we ensure, because so far it is only the 2N64 types of spontaneum. We found that 2N90, 2N72 are better than 2N64 in mobilization process. So the concentration is more on the high chromosome types of spontaneum. And also we gave more emphasis to the root mass, because whether it is for drought or for nutrient uptake or for mechanization, a strong root system is very much essential. So we have identified some of the spontaneum lines which have a very high root volume even under drought conditions, and there is very less biomass reduction under drought. These are being channelized into breeding program. And of course another species that the Ryanthus, these 2 species, namely Ryanthus aruntinaceus, known for its gigantic growth as well as multi-retooning efficiency and drought tolerance, and the Procerus types, which give very high proportion of red rot resistant parents, are now being utilized in breeding program. And in salinity studies with Ryanthus have found that there are many types which record higher net photosynthesis, low canopy temperature, and significant high upregulation of saline stress tolerant genes. So Ryanthus lines identified for high salinity are also used in breeding program, as well as in gene identification and used in genetic transformation. And GISH technique is helping us in identifying the true intergeneric hybrids, which are generally difficult to identify. These are with regard to using sugarcane with sorghum and sugarcane with maize. And we also identify several patterns of transmission of chromosome. There are 2N plus N, N plus N, N plus 2N, and different, and even androgenetic hybrids are also present. And there is a high level of pistoloidy representing, which limit the success of interspecific intergeneric hybridization. And also trait-specific hybrids are also included. And it's a success story that Ryanthus Procerus, we could get 2 co-varieties which have entered the ICAR, including the new source of Procerus, which is a non-cane forming type, but which give very high level of both drought tolerant as well as red rot resistant clones. And you can see there are many types within the R and MR category for red rot resistance, and cytologically also they have been characterized. And through molecular cytology, we have found that the BC2 clones having 5 chromosomes of Ryanthus Procerus, 5, 9 and 6 chromosomes of Ryanthus Procerus, showing that they are true hybrids. And also we are moving towards the chromosome additional lines, mainly with the Procerus lines. Again, value addition in sugarcane, which nobody has thought of. We have red flush that is robustum. After so many years, a few clones of sanguinium flowered. We made use of the opportunity, and now the BC2 hybrids are available, which are rich in antioxidants. That is sugarcane juice, all the goodness of sugarcane juice plus the value of antioxidants. Now that work is now progressing for value addition in sugarcane. Genomic approaches, so many studies carried out, and several genes have been identified, and even marker assisted selection. We expect that first the mass product will be available by 2026, and especially for drought tolerance. Red rot also, many markers have been identified from India and from elsewhere, from our work by Dr. Vishwanathan and others, and also from other group. So these are in the stage of validation, and our group is working on even doing marker-assisted selection for red rot. Then of course genomic selection. This is the result of a collaborative program with Sugar Research Australia, and with high throughput phenotyping also in coupled with this genomic selection, we are able to make a headway in this line. And this is the principle. I am skipping this, but how a genomic selection model can work. And even for sucrose, we found that there is 81% prediction accuracy using selected genomic selection models. And this has been even tried for red rot. The two groups of linked markers have been identified in the same chromosomal region. And using these two, the selection it's around 72%. So this work is under validation before publishing. And of course, to aid all these things, we have to have a very strong speed breeding program. Of course, our team has standardized the protocols for inducing flowering, because unlike other crops like wheat and rice, where it is well standardized, sugarcane is a short day plant, and the totally different conditions were needed in the case of sugarcane. And of course, transgenic technology, as it was told in Brazil and Argentina, we have a system already in place from 1999. But again, the restrictions, of course, 17 events are now ready for limited field trials, especially for Co-86032 with enhanced drought tolerance, and again, and also Co-238. Dr. Apunu and team, Apunu is already here, and his team is working on that. And several physiological parameters have already been studied, and several genes have been identified from our germplasm, along with the suitable promoter, which have been used in genetic transformation experiments. Several transgenics, even single gene as well as pyramided lines are available, both through agrobacterium as well as the bialistic method. And of course, gene editing, it's like in very infancy in India. Of course, there are reports of the canna canes from Brazil. We are now identifying with a few traits, maybe 3-4 genes, and some events are available in the case of ILD. This is again, the team is sitting here, Dr. Apunu and Dr. Vishwanathan. Some lines are now available which need to be validated. They are in the V0 line. And of course, sugarcane as a bioenergy. And though there are 4-5 strategies to improve the feedstock in biofuel, what we are mainly working on, it is lignin modification, that is altering the SG ratio. And for that already a gene has been identified, and that gene is being utilized. That is a ferulate 5 gene is being modified. And now the energy canes is another Erianthus arundinaceous derived clone. This has already been now in the PPP mode, already gone for commercialization, mainly for fodder, biogas, hydrogen, paper, particle and 2G. And this is the high biomass. You can see very high fiber as well as high biomass with this, and several, these have been commercialized, and biomass, and again the sugarcane as a platform for value added molecules. And here the vacuole of sugarcane where 90 to 95% any product can be targeted. We have a vacuolar targeting sequence which is in the very final stage of patent. We will be obtaining the patent very soon. So using that, our team is working on mainly for 3 genes, namely the glucose cerebrosidase, which is a rare genetic congenital disease, and also for insulin and interferon. And here this the downstream process standardization is in progress for the GXAAS, and it's again with an PPP mode with a laboratory called Trana Lab. And several of our technologies for product diversification are available, like value-added products from sugarcane. And our entrepreneurs are being given hand-holding as well as training in this aspect. And now this is the last slide. That is now era of collaboration. Researchers have to collaborate with industry and vice versa, so that the expertise for the product delivery, refining and value addition can reach the end users. And I thank also the ISMA for coming forward for a collaboration with SBI for a site-specific varietal identification on a pan-India basis. And with that, I thank everyone for this patient listening, and thanks for the opportunity. I also want to make an announcement that the 13th germplasm and breeding and the 10th molecular biology workshop of ISCT will be held in India, where for the very first time since the inception of ISCT, no conference is held in India. It's a rare opportunity. It will be held from 8th to 13th. It will be from 8th to 12th July 2024. It's a very rare opportunity. It's an international conference where many delegates will be coming from abroad. And this is the first time it will be hosted by Sugarcane Breeding Institute, Coimbatore, in collaboration with the industries. And I request all of you to be a partner, to come and participate in this one. Abstract has already been, submission has already commenced. And 29th February is the last date. Dr. Butterfield, who is sitting here, is one of the co-organizers. And I again welcome you to SBI for this very first ISCT plant breeding, that breeding and germplasm molecular biology workshop to be held in Coimbatore. Thank you very much.